Have you ever wondered how a muscle contraction works? Let's watch and find out. This is a model of a muscle. Muscles are made up of fascicles, which are bundles of muscle fibers. The endomysium is a layer of connective tissue that surrounds each muscle fiber, and a layer of paramysium surrounds each fascicle. Then, when several bundles of fascicles are grouped together, they are surrounded by a layer of connective tissue called epimysium. On the epimysium, mitochondria sit, which are filled with energy for cells. Going back to the muscle fiber, also known as myofibers, they have myofibrils in them. Myofibrils are made up of sarcomeres. Sarcomeres have thick and thin filaments that make up several different areas. The first area is the A band, which is dark. The I band is light. The Z line sections off each sarcomere. The H band is only where thick filaments lie, and the M line is in the middle where the thick filaments attach. A muscle contraction starts by having an action potential traveling down the dendrite towards the cell body. Once it hits the cell body, it transfers to the axon. As the action potential travels down the axon, it goes towards the muscle. Before it reaches the muscle, several things must happen. First, the calsequestrin, which houses the calcium, allows for the calcium to flow into the open calcium channels and move into the axon. Once in, the calcium pushes the synaptic vessels towards the synaptic knob. Once the vesicles reach the synaptic knob, they fuse with it and release acetylcholine. Acetylcholine is a neurotransmitter that stimulates an electrical signal. After acetylcholine is released, it moves across the synapse towards the motor end plate. The motor end plate is filled with proteins and is convoluted to allow for more surface area. The acetylcholine attaches to the proteins on the motor end plate and open to allow sodium to move into the muscle and potassium to move out. Once the muscle contraction is complete, an enzyme called acetylcholinesterase is released and stops the release of acetylcholine. And the muscle is now back at resting state. This is a structure of a muscle fiber. Muscle fibers are covered by a cell membrane called sarcolemma. As you travel along the myofiber, you will find little holes called transverse tubules or T-tubules. They are little depressions of the sarcolemma that push down into the muscle. The muscle fiber is filled with a cytoplasm called sarcoplasm. It also has a sarcoplasmic reticulum, which is physically separated from the sarcolemma and surrounds each myofibril. The introduction of calcium causes the troponin complex and the tropomyosin to shift, thereby exposing the myosin binding sites to the myosin head. The myosin heads are now in contact with the myosin binding sites. The myosin heads will move the thin filaments contracting the muscles. The ADP and phosphate, which causes the myosin head to move back, are left behind during the power stroke. The introduction of ATP causes the myosin heads to release the actin. Finally, the ATP is broken down into ADP and phosphate. Which causes the myosin heads to move back, ready for another power stroke.